actually he was not aware about the genes. So whatever traits or characters he observed that they are getting inherited from generation to generation, he called them as factors. Then all his findings he published in a paper called as Experiments in Plant Hybridization, where he actually explained the concept of hereditary unit. So hereditary unit means anything that is passed down from parents to the offspring. Then the term gene was actually coined by William Johansen, but the term genetics was given by William Bateson in 1905. So until then, actually, the Mendel's work did not get any kind of recognition. But when it got recognition after the rediscovery, so three scientists, your Carl Correns, Hugo de Vries, and Erin von Tesmark, they actually rediscovered or reworked on Mendelism, and then only Mendelism got popular. So as you already know, that the Mendelism is actually based on two important crosses conducted by Mendel, that is the monohybrid and the dihybrid cross, for which actually he chose the pea plant. The pea plant, why he chose the plant particularly? Now, the plant had certain means interesting characters, like it had very short generation time. That means within a means कम टाइम में बहुत generation of plants you can observe. So if you are observing something that is passed down from generation to generation, a short generational time plant will be appropriate to study the character inheritance. Then he observed that in that particular plant there were very distinct contrasting characters. Means you can observe here whatever characters he had chosen, they were very different from each other. Like if we see the seed color, one color was yellow, the other color was green. So there were completely contrasting characters which, okay, anyone put any questions or something? Okay, fine. So as you can see, so there were different type, means very different colors or different character that can be easily distinguished from one another. Like yellow is a very different color, green is a very different color. So it was easy to identify. Then the plant was able to be self-fertilized as well as cross-fertilized, which is one of the most important aspects to study how exactly the gametes are being formed and how they are combining in the zygote. Then it was easy to cultivate. It had low maintenance and it was very easy for Mendel to cultivate this particular plant. So next, if we move to the monohybrid cross, so mono, it simply means one. So as he was taking or he was considering only one character at a time, the test was called monohybrid cross. So as you can observe, he only considered one character, that is the height of the plant. So he took two contrasting character plants. One was absolutely tall or pure tall plant. The other one was dwarf or pure dwarf plant. So as we can see, since the tall plant, it had only one type of allele, so only the capital T will be inherited to the gamete, and from the dwarf plant only small t. So in the F1 hybrid, what could happen? Now the T and T will combine and it will form a heterozygous offspring. Then if this was self-fertilized, the capital T small t, so we can have two different type of gametes in each plant, and if they combine, we will have these kind of genotypes. So one will be capital T both, capital T with small t, then small t. And if we look into the phenotype, so phenotype means the how the character is appearing or how we are looking into it. So the phenotype here is two types. One is tall, another is dwarf. So this plant, all the three, they exhibited the tall character. Only this plant exhibited the dwarf character. Then if we look into the genotype ratio, so genotype ratio means how the alleles are being combined in different ways or how they are arranged in a particular diploid organism. So each gene will be consisting of two alleles as we are diploid individual. So we will have in each genotype, we will have two different type of allele. So the allele might be same, like you see in the first offspring, both the alleles are exactly same. So this is a condition of homozygosity. And here if you see, both the alleles are different. One is capital T, other one is small t. So here it is heterozygous condition. Here, both the alleles are recessive or small, but both are similar. So again, this is a condition of homozygous. So the phenotypic ratio is 3 is to 1, and the genotypic ratio that was from Mendelism was 1 is to 2 is to 1. So this is about the Mendelian monohybrid cross. So what he actually concluded from the monohybrid cross? Now, if you observe to the result of the cross, whatever he conducted, in the parental generation, there were two different type of character. One was tall, other was dwarf. But in the F1 offspring, if you see, every offspring became only tall. So the dwarf character did not appear in the F1 generation. Then when the F1 was self-crossed and the F2 generation was obtained, as you can observe, there was again reappearance of the dwarf character. So what kind of principles he deducted from the monohybrid cross? Now, two major principles were deducted. One was the principle of dominance. Okay, so in heterozygous condition, one of the character will be always dominating over the other one. Okay. So as you can see, one of the character will be only expressed over the other one. So here the tall was the dominating character and the dwarf was the recessive character. Okay, I can see someone raising the hand. So do you have any issue? I think you can unmute and speak. Or you can simply write it in the chat box. I will read and I will respond to your queries. Okay, so as we observed, so here then the what was the first law? The first law or first principle was the law of dominance. That okay, who is Subha Sri Lanka? You have any problem? Because I can see you have raised your hand. Okay, so so this capital T, it is actually it is not allowing the small t to express itself. So this is the dominant character, this is the recessive character, the small t. And the other thing was the law of the law of segregation. That means during the gametogenesis, the gamete will receive only one copy of the allele. So when in the heterozygous parent there was capital T, small t both, but what about others? You all can hear me clearly, right? I am loud and clear to all of you.
Yes, ma'am.
See, most of the students can means they can actually hear me properly. So if you are having any problem, the problem might be from your side. So when if anyone is facing any kind of issue, audio or video, please try to log out and log in again. Okay, because if the problem will be from my side, everyone will face it. So if one or two learners, if you are getting any problem, then please try to log out and log in again. Okay. Okay, fine. So here the second law was the law of segregation. That means during the gametogenesis, each gamete will receive only one copy of the allele. Okay. Then let's move to the next cross, that is the dihybrid cross. So in the dihybrid cross, as you know, the two contrasting characters were used simultaneously. So he chose the shape of the seed as well as the color of the seed. So the contrasting characters were actually a slight mistake is here. It should be green. Okay, it should be green and wrinkled. But here it is appearing yellow and brown. So it should be green and wrinkled like this. Okay, this should be here. So here, as you can see, so two different characters he chose. That is why dihybrid cross. So yellow and round. So in the parental generation, there was yellow and round, as well as the opposite character, that is the green and wrinkle. So again, like the monohybrid cross, the F1 results were consistent. That means all the F1 generation exhibited yellow and round. So from this, we can predict that the yellow and the round character are the dominating character over the green and wrinkle. But now, when the F1 was self-crossed, as you can see, two new different combinations were developed. So what are they? As you can see, one is here, that is round in a yellow color. Okay, so earlier it was round with yellow, but now you have round with green. Similarly, the second new character is wrinkle with yellow, but in the parental generation what we had green with wrinkle. So these two new combinations they occur because of the independent assortment of the characters which are located on two different chromosomes. Okay, so for what was the deduction from the dihybrid cross? Now it was the law of independent assortment. That means when a pair of contrasting characters are segregating from each other, then they will segregate independent of each other. So how it occurred? Now if you observe here, during you have studied the cell division. So in the cell division during anaphase, the means paternal and the maternal, or we can say the two different chromosomes are pulled to two different poles. So if you see, if the alignment is like this, so here this is capital G, okay, and this is the small g. This has capital W, this has the small w. Okay. So if the alignment is something like this, so what you observe, the capital G and the capital W, they are moving to one pole or they are moving to one gamete. The small g and the small w, they are moving to other gamete. But somehow if in the cell, the chromosomal arrangement is like this. The capital G has been aligned with the small w, the small g has been aligned with the capital W. So then you will have a different combination like this. So this new different combination is known as a recombinant. Okay. So this occurred in case of Mendelian cross because of the location of the characters in two different chromosomes. So the law what he derived here is law of independent assortments. Means the assortment or the movement of the color gene is completely independent of the movement of the shape gene. Okay, because both the genes are located on two different chromosomes. So that's how you are getting these new combinations. Okay, so this could be your yellow and round. This could be wrinkle and green, but here you are getting a yellow with green and wrinkle with a yellow shape. Okay, so these two are the new recombinant characters. Then next, if we move to the trihybrid cross, like in the dihybrid cross, you have different type of genotypes and phenotype ratio that you are obtaining. Similarly, you can do it for a trihybrid. So tri means what? There will be three characters considered at the same time. So like in monohybrid, we have the phenotypic ratio 3:1, the genotypic ratio 1:2:1. In case of dihybrid, we have it, the phenotypic ratio is 9:3:3:1. Okay. So all the dominant character will occur 9 times. This new recombinant character will occur 3 times. Again, the other recombinant character will occur 3 times and the recessive character will occur only once. Similarly, the genotypic ratio will be 1:2:1. Again, 2:4:1. Sorry, 2:4:2, and again 1:2:1. Okay. So same can be done for trihybrid cross also. So in trihybrid cross, we will be considering 3 characters at a time. As you can observe here, there is the shape. Sorry, the first there is the color yellow and green. Then the shape round and wrinkle. Okay, then the height, the tall and the short. So let me draw a simple thing for you, so it will be easier for you to understand. Like in dihybrid, what we can say, if we consider like this. Okay, I'm just using the blank sheet from a PowerPoint. Just let me know if it is visible to you or not. In genetics, actually, a lot of drawing is required. So the blank slide is visible to all. All can see.
Yes, ma'am.
Yes, ma'am.
Is it
Yes,
blank
ma'am.
Yes, ma'am.
slide? Okay.
Yes.
So if I write like this, capital Y, small y, okay, capital R, small r. So if I am self-crossing it with again another genotype, capital Y, capital Y, capital R, small r. So just tell me, if I consider this only, isn't it same as the monohybrid cross? Capital Y, small y. So it is same as the monohybrid cross. So what kind of combination you will get from this? You will have a capital Y, capital Y. Okay. Then you will have a capital Y and small y combination. Then you will have a small y, small y. Then again, if you consider this RR once more, so this is also again kind of monohybrid cross. So again, what kind of genotypes you will expect here? You will have one capital R, then capital R, small r, and small r. So since there means both the genotypes we are considering, so they will be actually kind of this YY will combine with this YY and RR. Similarly, this RR will also combine with this YY as well as this RR. So here, let me use a little bit. So here also what we will get. So here also we will have capital R, then capital R, small r, small r, small r. Okay. So now if you remember this monohybrid cross Y and capital Y, small y, the frequency of capital Y and capital Y will be 1. Okay. The frequency of capital Y with small y, okay, both it will occur twice. Okay. So small y from this, capital Y from this. So y will be 2 times. So this frequency will be 1, this frequency will be 2, and small y will be only 1. So similarly, you can use the same frequency here. So here it will be 1, then RR will occur frequently once, then capital R, small r will occur 2 times, small r will occur once. So if you multiply it, y, then r, r. So what is the frequency? The frequency will be 1. 1 multiplied with 1. Then if you see capital R and small r, then the frequency will be 1 into 2, that is 2. Okay. So in this way, if you calculate the ratio, you will get 1 is to 2 is to 1. Again, 2 is to 4 is to 2. Again, 1 is to 2 is to 1, which you can observe in this slide. So if you look at the genotypes, the RRYY is occurring only once, and the capital R, small r, capital Y, small y, it will occur 2 times like this. So if you arrange the genotypes, you will get that the genotypic ratio will be 1 is to 2 is to 1, again 2 is to 4 is to 1, again 1 is to 2 is to 1. Similarly, here the for the trihybrid cross, the phenotype has been written like this. Because if you observe the means only the color will be in a ratio 3 yellow is to 1 green. Similarly, the shape will be again 3 round and 1 wrinkle. 3 round and 1 wrinkle. Everything will combine with the color again. The shape will again combine with the color. So like this, the height will be also 3 is to 1. Okay. So if you multiply them, what you will get? That 27 plants will be yellow, round as well as tall. Similarly, 9 will be yellow, round and short. Then 9 will be yellow, wrinkle and tall. 9 will be yellow, 3 will be yellow, wrinkle and short. So this is a method which is called as the fork line method. If you Google also, you will get the same means how exactly it is done. So it is like if you are going for more than dihybrid cross, you can use this method to predict what will be the possible genotype. So for the trihybrid cross, what is the phenotypic ratio you will get? Now 27 is to 9 is to 9 is to 3 is to 3 is to 3 is to 1. Okay. Then coming to what is mean by test cross. So test cross means if you look at the So if you look at the it is still visible, right? The slide.
Yes, ma'am.
Okay, so if you see, if I write it like this, capital P, this will also give you a purple flower. If I write capital P and small p, then that will also give you a purple flower. So how you will predict which, actually what is the genotype of the purple flower? So for that purpose, if you cross it with a white flower, so white flower, it will have only small p genotype. That means it will never have any color, and this will also always have the recessive allele. So this parent will always give you a small p allele. Now, depending upon what is the possible genotype of the purple flower, you can observe two different outcome of the test cross. So, if the purple is homozygous dominant, all the gametes will only have the capital P. And when this capital P will combine with the small p of the recessive parent, all the offspring will appear purple in color. But by chance, if the purple flower is having a capital P and the small p, then it will have two types of gametes, or half of the gamete will have the capital P and half of the gametes will be having the small p. So when they will combine with the small p, you will have heterozygous purple, or you will have white. So there will be a ratio of 50-50, 50% dominant and 50% recessive, or the ratio of the dominant is to recessive phenotype will be 1:1. So since by crossing a unknown genotype to a double recessive parent, you can actually predict the genotype of the unknown, means unknown individual. This kind of test cross is known as the test cross. Okay, so the test cross ratio for a monohybrid test is always 1:1, and for a dihybrid it will be 1:1:1:1. That means all the characters will appear in equal proportions. Then coming to the next thing, that is extensions of Mendelian principle. So up to this, we had the Mendelism. That means there was no linkage, no crossing over. We are assuming that every gene is occurring in two alleles, and there is concept of dominance and recessiveness. But some things could go beyond that. Like if you observe in this first diagram, you are crossing a purple flower with a white flower, and all the F1 are coming out as purple. So this is a clear case of dominance, which is following the Mendelian principle. The purple is dominant over the white. But sometimes what you will observe, if you cross a red flower with a white flower, so what we should expect, that all the flowers should appear red, because red is supposed to be the dominant color. But in contrasting to this, what you will see, all the flowers will appear pink. So which is neither red nor the white color. So this is a condition where the red is trying to dominate over the white but cannot completely dominate it. So this is an example of incomplete dominance. So for the incomplete dominance, the genotypic ratio is same as the phenotypic ratio. For the F2 generation, okay, because the only the homozygous will give rise to in incomplete dominance, what you will observe, now all the characters or all the offspring will be having a intermediate character between both the parents. Neither of them will have the, means none of them will have the dominant parent character, none of them will have the recessive parent character. So they will have a intermediate character, and 100% of the offspring will exhibit the same character. But again, there is another concept which is called as the co-dominance. So co means together. So here the dominance effect is there, but it is incomplete, not complete. But in codominance, what you can observe, now in codominance, both the characters will be equally expressed. So here, if you see, or if you are crossing a white color cattle with a red color cattle, you can see there will be patches of both red and white. So this is an example of codominance. Then comes the multiple allele. So till now we just saw that most of the alleles will be occurring in pairs, and there will be two possible characters for any two possible allelic forms of any character. Like for height, there is either capital T or small t. Okay, like for the color of the flower, there is either purple or white, like this. But in some cases, what you can observe, then there will be multiple alleles for the same character. So, like if you see here, the in the rabbit, the coat color, it can be governed by four different alleles. One is the capital C, that is coding for a brown fur. The other one is a chinchilla, that means it will have the actually white fur, but at the end there will be black tip, like this. If this is the fur, at the end there will be black tip, so it will be appearing more kind of blackish. Then you have the Himalayan allele, where the rabbit is appearing almost white except at the extreme positions like the ear, the tail, the foot, etc. Except this, the rest of the body will be white. Then the last one is albino, that is completely white fur. And in case of multiple alleles, there is an order of dominance like this. The most dominant character is the brown color, followed by the chinchilla, followed by the Himalayan, followed by the albino. And here is a small formula how to predict the number of genotype in case of multiple allele. In case of multiple allele, the genotype can be calculated by a formula n into n plus 1 divided by 2. Okay. One second, let me use the mouse. Okay. So it is n into n plus 1 divided by 2. So like in this case, we have four different alleles. So what will be the possible number of genotypes? The possible genotypes will be 4 into 5 divided by 2, that is 10. So for this particular character in the rabbit, you can have 10 different possible genotypic combinations. Similarly, here also if you observe the eye color in Drosophila, so there are four different type of alleles. One is this E1K. So that is actually coding for wild type. Then you have the apricot. Then you have the honey color. Then we have the white. Okay. So, or it is simply colorless, we can say. So here if you see, the E1 is actually controlling the wild type, and the E2 is coding for the apricot, then E3 for the honey color, and E4 is coding for the white color. So if you observe, there are how many genotypes are possible? 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay. So this is a small tree to calculate the number of genotypes. So one best possible example of multiple alleles is our blood group. Okay, so in blood group we have actually three different type of blood group in ABO blood group system. One is A, B, and O. So if you look into the possible genotype, so what should be the possible genotype? That's 3 into 4 divided by 2. That means 12 divided by 2, 6. So that you can see here clearly, we have 6 different type of genotypic combination. One is IAIA, IAIB, then IAIO or I0. This is okay, then okay. So this is the same. Then IB, then IB and 0. Then last one is IOIO. Okay. So these are the different combinations: 1, 2, 3, 4, 5, 6. So 6 possible genotypes are there for our human blood group system. So if you know the molecular reason behind this blood group, if you see in this case, in case of blood type A, okay, so the RBC has antigen A, and in the blood or on plasma, we have anti-B antibody. Okay. Then similarly for the blood group type B, we have the antigen B on the RBC, and in the plasma we have anti-A antibody. And for the blood
And
group
slide
AB,
has only
we have both
slide
the
has
antigen A and B on RBC, but no antibody in the plasma. And for type O, if you see, there are no antigens on the surface of RBC, but both anti-A and anti-B antibodies can be found in the plasma. Now look into the molecular mechanism, how exactly the blood group is being defined. So if you see, there is a H substance. Okay, so the H substance precursor is actually governed by another
मैम
gene,
स्लाइड चेंज हो गई
capital. Okay, so here
एक्चुअली
if you see the means H
मैम
precursor is actually
मैम
yes, yes. Any problem?
स्लाइड चेंज होनी
सेम अच्छी मैम चेंज
Okay, okay. The slide has not been changed.
यस
Okay, okay. I'm sorry. Uh, it has changed now.
यस
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 मैम
मैम
Okay.
यस मैम
Okay, you can always interrupt me and tell me if there is an issue. Okay, so actually, if I don't means go for slideshow view, I cannot write anything, and without writing, it is difficult. Okay, to explain. So I hope now, if I'm writing, also you can see that, right? The pen marks and all.
नो मैम
Uh, okay, okay, fine. Then you just concentrate if it is difficult to write. So just look at this means conversions. Okay, so there is a H substance precursor, the FUT1 allele that directs the addition of fructose to the H substance precursor. So this is the primary means change during the synthesis of the blood antigen. So by default, everyone will synthesize this O type blood group. Then there are two types of enzyme that will add to different functional groups. So if you see in one of the, if the A antigen has to be there, so there is an enzyme that this galactosamine kind of, I'm just forgetting the name of the substance. Somehow you observe this is the chemical group that AC gal NH. Okay, so this group is added here, here and here. So except this, the blood type A and B both are same. So only in antigen in individual who have the blood group B, they will have the addition of a galactose here, and in the blood group A, instead of galactose, they will have this compound. Okay, so that is the only difference which is creating blood type A and B. So in the individuals where the A and B enzyme both are present, that means the galactose transferase is also present and this substance transferase is also present. It will add both the antigen to the RBC. So they will have the blood type AB. And in the individual where they do not have either of the enzyme, it will simply result in no addition or no modification of the bases, and it will remain as O type blood group. So here comes another interesting fact. That means if you observe here, whatever antigen is present, that antibody is absent in the plasma. Here antigen A is present, so anti-A antigen antibody is absent here. But in case, what happens in case of blood transfusion? So transfusion means what? When the blood is transferred from one person to another person. So if the recipient is Rh negative, but the donor is Rh positive. So if the recipient is Rh negative means it will have the antigens, sorry, it will have the Rh antibody. Okay, anti-Rh antibodies are present in the recipient. So when the donor with a positive blood group is transfused into the recipient, the antibodies will start attacking the antigen, because always the antigen and antibody will have a interaction, or they will have a affinity for each other. So once the antibody starts attacking or interacting with the Rh antigen, so it will lead to the lysis of the blood cells. So whatever blood is donated to the recipient, most of the RBCs will be destroyed. So this leads to a condition called as the Rh hemolytic anemia, because loss of RBC means loss of blood, So this will result in the anemic condition in the recipient. That is why when the blood transfusion is happening, you must ensure that both along with the blood type, that means AB whatever is there, the RH compatibility must be there. Okay, so no donation should be occur between a RH negative recipient and a RH positive donor. Now another thing happens here, like in case of your pregnancy, in case of your development of the fetus, if by chance the mother is RH negative and the father is RH positive. So if the father is RH positive, the usually the positive is the dominating condition over the negative one. So the fetus or the child that is growing inside the mother will have the RH positive blood type. Now the mother is RH negative, so the mother blood or the maternal blood will have the anti-RH antibodies. So what they will do, the blood of the fetus and the mother will come in contact with each other. So the anti-RH antibodies of the mother will attack the RH positive RBCs of the fetus. Okay, so this might lead to the abortion of the child, a condition called as erythroblastosis fetalis. Okay, so the child will develop the hemolytic anemia and it might also lead to the death of the child. So a condition where in a couple where the female is RH negative and the male is RH positive, they might get their first child because the development of the anti-RH antibodies and the interaction with the fetal blood may take some time. But once the anti-RH antibodies develop, the second child will never be possible because of a condition called as erythroblastosis fetalis. Okay. Then next comes the lethal allele. So lethal allele means a allele that will result in the death of the individual or in the death of the organism that possesses the gene. So just let me know if I'm drawing something you can observe, means you can visualize or not. Like this. Is it visible, the drawings?
No, ma'am.
No, ma'am.
Okay, this is actually a very difficult
No, ma'am.
question. Okay, fine. I don't know why it is happening. Okay, if I'm drawing here, it is visible, right?
Yes, ma'am.
Like this. Okay.
Yes.
Okay,
Yes,
fine. So we will
but yes.
continue like this only. Okay, but it is going fading away so soon. Okay, fine. So just observe here. As you can see, there are two different alleles that is controlling the coat color in the mice. One is the capital A, the other one is capital A with Y. Okay, like this. So this is actually coding for a yellow color coat and the capital is coding for a agouti, and that is a brown color mouse. So if the mouse is capital A, it will develop the brown color. Okay. And if the mouse is capital AY and capital A like this, means it is a heterozygous condition. Then also it will develop yellow color. But if the mouse will be both capital Y, okay, then the Y actually in homozygous condition it is lethal. That means no mouse with both the Y allele will survive. So this Y allele is known as a lethal allele. Okay, so here the normal phenotypic ratio, which should be 3:1, will change is to 2:1, because if you compare it with the monohybrid cross of the Mendel, that is the capital T thing, so this capital T will not survive, means the first homozygous dominant condition will not survive. Okay, so only the heterozygous will give you the dominant character and the homozygous recessive will give you the recessive character. So here one of the dominant phenotype will be lost and the phenotypic ratio will change to 2:1. Then next, coming to some examples. Okay, examples of Certain examples of lethal alleles, like in our case, we have examples of like sickle cell anemia, thalassemia, Tay-Sachs disease, cystic fibrosis. Okay, so these are some of the mutations in homozygous recessive condition. They will be lethal to the organism. So here the ratio will be 3:1. That means if two heterozygous parents or two heterozygous parents will be means if we made two heterozygous genotype, the homozygous recessive genotype will be always dying. Okay, similarly, there could be certain dominant lethal like Huntington's disease. Okay, then achondroplasia. So these are some of the diseases in where in what condition the homozygous dominant will be lethal or it will be dead. Then certain conditions are sublethal. So sublethal means usually you will not die of this genotype, but if some problem occurs, then it will lead to death. Like in case of hemophilia. So in hemophilia, what happens? The clotting factor is actually lost. Means if some cut or minor injury occurs to us, usually the blood will cut. Okay, if we accidentally cut or if we bleed, it will be stopped after some time. But those individuals who have hemophilia, the clotting factor will be lost, so the blood will never cut. So even the minor wounds can lead to in death also. Then there is conditional lethal. So conditional lethal means the gene is lethal in certain conditions and it is absolutely normal in other conditions. So example like fabigism. So fabigism means there is a kind of what we can say means a kind of pod or a kind of lentil actually. So lentil means like moong dal kind of thing. So that beans. Okay, so the bean, if the individual who have a G6PD deficiency, okay, glucose 6 phosphodehydrogenase. So there is an enzyme called this G6PD. So the individual who have a deficiency in this enzyme, so if they consume or if they eat this faba bean, they will be dying because of hemolysis. Why? Now this faba bean, they will create a number of oxidative stress on the RBC, and the RBC will be simply burst or will be ruptured because of the presence of the faba bean release. So if this G6PD enzyme is there, they can neutralize the oxidative stress that is developed by the faba bean. But if the individual is deficient in that particular enzyme, then it will die. So that means the lethality is depending upon the condition. If you are consuming that thing, then it is lethal to you in that condition. If you are not consuming, then it is not lethal. Now coming to why some things will be recessive and why some things will be dominant. Okay. So the recessive condition will be there because of the loss of function mutation. So like if you observe here, as long as both the copies are normal, the gene product will be released and the normal function will be exhibited. If one of the gene is lost or mutated, then also one copy is there to compensate for the lost gene. So when you can observe the disease, now when both the genes are mutated. So when both copies of the gene will lose the function, then only the lethal condition will develop. So this recessive character is developing because of the loss of function mutation. Similarly, the dominant condition will develop because of gain of function mutation. So gain of function. Okay. Now I guess everyone is able to see the slide and all, right? That is no problem. Yes, that is quickly disappearing. I'm really sorry regarding that, but that is how I think the system is working. Whatever I'm writing, it is immediately getting. Okay, now it is saying no, it is also going up. Okay, so I'm trying to explain, so please try to cope up with that. I cannot do anything regarding the system. Okay. Okay, so why some cases will be dominant? Now, when there is a gain of function mutation. So if you see, this is the normal gene product. Okay. So if one of the gene is also getting mutated or one of the allele is getting mutated, then also the mutant effect can be seen along with the normal one. So if even if one copy is mutated or both the copies are mutated, you can still observe the mutant effect over here. So that is why these characters will appear dominant in nature. So if there is a gain of function mutation, it will result in a dominant character. If there is a loss of function mutation, it will result in a recessive character development. So next, we can see the example of the sickle cell disease. I hope everyone knows about the sickle cell anemia. So in sickle cell, if you see, this is the normal shape of the RBCs. Okay, that is oval shape. But if there is a slight change in the beta globin gene of the hemoglobin, as you can see, there are actually it consists of 146 amino acids. Okay, so here at the sixth position, if you observe in the normal hemoglobin, there is glutamic acid. Okay, but in the sickle cell hemoglobin, the glutamic acid is replaced by valine. Okay, so due to this small change, the shape of the RBC will be changed from oval to sickle cell like this. Okay. So how you can observe by looking at the the Western blot analysis of the protein or the gel duct of the protein. Now, if you see the normal protein, it is actually a long protein and it cannot be cleaved or it cannot be cut at any point. Means no restriction enzyme can cut it. So if you see the weight of the protein is quite high and you can observe this is the band that you will observe. Now, if you if someone is heterozygous, means having a normal copy as well as a mutated copy. So the mutated copy will be actually smaller in size. So you can observe two bands like this. And if you observe only a single small band, that means both the copies have been mutated. Okay. So this is the gel profile of the normal heterozygous as well as sickle cell hemoglobin. Okay, sickle cell beta globin chain. Then now coming to gene interaction or epistasis. So here, if we observe, actually it is
Ma'am,
very.
can I have a question?
Yes, you can ask.
Please show the previous slide.
Actually, it is very difficult without writing. Explaining the genetics is very difficult. Okay, you can put your
Madam,
question.
the electrophoretic pattern, the
Yes.
electrophoresis direction. Would you please explain a little bit about this?
Okay, see, actually, this is a technique. Okay, this technique is based on the weight of the molecule that whatever you are analyzing. So, the heavier the molecule, the slower it will move. Okay, because it is difficult for a heavy molecule to move much farther. So, if you observe here, means the molecule is heavy, it is unable to move much farther, or it cannot move very far. So it will stay somewhere near where the sample has been loaded. So this is representing a normal globin gene. Okay, normal globin gene. But if there is a mutation, this because of the substitution of the glutamic acid to the valine, this actually creates a restriction site in the gene, beta globin gene actually. So what will happen in that case? The restriction enzyme can cut it. So if it can be cut means smaller fragments will be there. So in case of heterozygous, where there is one normal copy, you can see one normal copy will move and stay here. The other copy, which is mutated, that is smaller in size or that is a smaller fragment, can move a little farther. So this is representing a heterozygous condition, a normal globin gene and a mutated globin gene. Okay. And if it is mutated in both the genes, means both the copies will have valine instead of glutamic acid. So it can be cut for both the copies. So it will move very farther because this will be now smaller in size and the fragment size will be small. So it will move far away from the sample where it has been loaded. So this pattern will give or it will indicate that this is a sickle cell condition. Okay. So these are
Thank
for
you, ma'am.
diagnosis purpose actually. Okay, fine. So then comes the epistasis. Okay, so epistasis means till now we observe the allelic interaction. That means where two alleles are actually means interacting among themselves, like the capital T, small t. They are alleles of the same gene. The capital T is dominating the, or it is masking, or it is suppressing the expression of the small t. So these kind of interactions are called as allelic interaction. But in some cases, as we will see, so there could be yes, I will try. So don't worry, okay? Because these things, if you do not understand, means there is no point of then taking the class. The epistasis, I think this needs to be explained. So I will try my best to move faster. Okay. So, okay. So here, if you see what is epistasis means, so epistasis is actually a non-allelic interaction. That means here, alleles of two different gene will be affecting the outcome of each other. So these are the examples of certain type of epistatic interaction. One is complementary gene interaction, then supplementary gene interaction, okay, then duplicate factor, inhibitory factor, polymeric or additive factor, etc. So now if you observe the normal dihybrid phenotypic ratio that we had 9:3 to 3:1 will be now changed. So it will be 9:7, 9:3 to 4, 12:3 to 1, and so on. So let's see how exactly it is happening. So I will give you two examples. Okay, so as you can observe here, so here if you look into the pathway, so there is a colorless product which is converted into a colorless intermediate by the capital C, and the colorless intermediate is converted into a purple pigment by the activity of capital P. So if you hide this part, okay, so what we should expect? Now wherever there is a capital P, we should expect a purple color. But if you observe carefully, the effect of capital P is actually influenced by this capital C. So if you see here, okay, so just okay, here if you observe, so here there is a capital P. So by the law of dominance, we should expect that the capital P will give a purple pigment. But what phenotype you are observing here? The phenotype is appearing white. So this is an example of epistasis where the capital P or the effect of capital P is inhibited or masked by the effect of capital C. So this is an example of a duplicate recessive epistasis, and the modified ratio is actually 9:7. So you just need to remember the modified ratio. Okay. So this is an example of duplicate recessive epistasis. Similarly, we have other types like here you can observe. Okay, so here this capital W is actually inhibiting the conversion of white to yellow, and the capital Y is inhibiting the conversion of yellow to green. So when we have a capital Y, we should expect a yellow phenotype, and when we have a capital W, we should expect a white phenotype. So if you observe wherever there is a capital W, you are getting white. So there is no problem in that. But if you see here, there is a capital Y, but still instead of appearing yellow, it is appearing white in color. So why? Because the capital W is inhibiting the conversion of white into yellow. So the capital W is actually affecting the expression of the capital Y. So this is an example of a dominant epistasis. Because both W and Y are dominant alleles, although they are not related to each other, but still, since they are part of the same metabolic pathway, they are actually affecting the expression of each other. So these kind of non-allelic interactions are known as your epistatic interaction. So there are various other types also. You can go through that. Okay. So I have listed few here, and also they are present in your material. So I guess if you go through them, you can actually find out what is happening. Then next we have the polygenic inheritance. So poly means many. So as we saw in case of monogenic trait or Mendelian trait, only the one there the character is controlled qualitatively. Means if there is capital T, Means if both of them are capital T, then also the plant height will be tall. And if the genotype is capital T small t, then also the plant height will be tall. And the height of both capital T as well as capital T small t will be small, same. So here the quality of the tea is mattering. Okay. But in certain cases, what you can observe, now as the number of dominant genes will be more, the character will be more. So if you observe here, as here there are 4 dominant genes, so each gene is actually contributing to certain character. So the phenotype is darkest red here. And as you observe, as the number of dominant gene is decreasing, the character is also decreasing. So from dark, as here the number of dominant gene is only 3. So here the character is darkest red to dark red. Here the only 2 dominant genes are there. So the character is medium or the color is medium. Here only 1 dominant gene is there. So the color is quite light. And here, since no dominant genes are present, the color is absolutely nil or it is actually 0 color. Okay. So this is an example of polygenic inheritance where the alleles are added. This is also called as additive gene. Okay. So the dominant characters, they actually add. The higher is the number of dominant allele, the more is the expression of the character. So this is actually more in quantity in contrast to quality. And in contrast to Mendelian ratio, where the phenotypic ratio is 9:3:3:1, the phenotypic ratio for the F2 generation in polygenic inheritance will be 1:4:6:4:1. Okay, so this ratio only you need to remember. So next we have the same thing. If both all the dominant genes are present, the color will be like this, bright red or wild type. And if one of the dominant gene is lost, the color will be scarlet. Okay. And if this dominant gene is present, this one is lost, then the color will be brown. And if none of the dominant genes are present, the color will be white. Okay. So this is also an example of qualitative gene. Sorry, quantitative gene or polygene. And this is also example, it's same, means as you can see, if the number of dominant alleles will be more, the character will be walnut. As the number of dominant gene is decreasing, the character or the phenotype will also change accordingly. The same thing here also. Now comes what is meant by synthetic lethal. So synthetic lethal means this is actually mostly used for cancer therapy. So in a wild type character or in a wild type cell, if you see, this is a normal gene and means 2 different genes are there. So the cell is actually healthy. But if one of the gene is being mutated, okay, so like as you can see, there is a BRCA1 gene. So this is actually a marker gene in the breast cancer. So the mutation in the BRCA gene actually leads to development of the breast cancer. So in a cell where in a cancer cell, if the BRCA gene is only mutated and the P gene is this RPE1 gene, that is PARP gene, if it is normal, then the cancer will run vigorously or it will grow vigorously. But if this gene is also mutated, means simultaneously 2 mutations are done. The Q is also mutated, the P is also mutated. Then the cell cannot survive. So this is called a synthetic lethality. You are making the second mutation purposefully, or you are synthesizing a second mutation to disrupt the livelihood of the cell. So this is what is called synthetic lethal. Then there is the gene suppression. So gene suppression means where a second mutation can suppress the effect of the first mutation. So this is a case of beta and alpha thalassemia. So if both the cells are normal, then the cell is having a normal phenotype. So if one of the cells becomes mutated, means there is the beta globin chain got mutated. So this will lead to disease. Okay. But if the beta gene is mutated and also the alpha gene is mutated simultaneously, so they will be actually compatible and the cell can have a normal phenotype. So here a second mutation or a suppressor mutation in the S is actually beneficial or is actually suppressing the mutation of the first one. So this is what is called genetic suppression. A second mutation can suppress or can inhibit the effect of the first mutation. Then comes pleiotropy. So pleiotropy means when a single gene can affect multiple phenotypes. So for example, if you see, this is a normal cell, okay, this is a sickle cell. So sickle cell, as we have already discussed, the RBC will have a abnormal shape. So because of this sickle cell, there will be so much characters. Like there will be anemia, there will be heart failure, there will be pain and fever, there will be brain damage, etc. So a single mutation in the HBB gene, that is the beta chain of the hemoglobin gene, so many characters or so many phenotypes are developing. So this is what is called as pleiotropy. Then next we have sex-linked inheritance. So if you have studied this Drosophila experiment that was done by T.H. Morgan, so he actually, what he observed, now a few characters, they can be actually transmitted or inherited through the linkage to the sex chromosome. So if we observe, we have two different types of chromosome. One is the autosome, the other one is the sex chromosome. So in case of humans, if we see, we have 22 pairs of autosome. That means 44 autosomes are there, which are same in both male and female. And in case of males, we have sex chromosome, that is X and Y. In case of female, we have homozygous X chromosome, that is X and X. So normally the somatic characters are inherited through the autosome, but in some characters are there which can also get inherited through the sex chromosome, that is either X or Y. So that is called as a sex-linked inheritance. Like we discussed the hemophilia in case of human beings is actually inherited through the X gene. Okay. Similarly, the color blindness. So these are actually somatic characters. So although they are somatic characters, still they are inherited or they are actually present on the sex chromosome, that is X chromosome. So this thing is called as a sex-linked inheritance. Okay. So it was discovered by Morgan in Drosophila. So what was the experiment actually? Now when he crossed a red-eyed female with a white-eyed male. So what he observed? Now all the progeny, irrespective of male or female, they had red eye color. That is the color of the mother. But when he went for a reciprocal cross means he just exchanged the character between the mother and the father. So now the mother was white-eyed and the father was red-eyed. So he observed what? Now only the daughters had red eye color, but all the sons, they had white eye color. So what he concluded from this? Now definitely the color that the gene that controls the eye color in Drosophila is actually located on the X chromosome. Since the X chromosome for the mother is distributed to both son and daughter, when the mother had red eye color, all the progeny had red eye color. But in case of daughter, only the daughter is receiving the X chromosome from the father. The father is not contributing any X to the son. Only the Y is given by the father to the son. So when the father had red eye or only one X chromosome is present in the male, so the X is donated to the daughter. So only the daughters had the red eye color. And since mother had the mutant X or the white-eyed X, so all the sons, they had white eye color. So this is what was explained by Morgan as sex-linked inheritance. The somatic characters inherited by the sex chromosome, that is primarily the X chromosome. Then comes a concept of sex-limited inheritance. So limited inheritance means what? Now some characters that are limited to only one gender of the organism. So like in case of humans also, if you observe, the development of beard, the development of breast, it is limited to one particular sex only, either male or female. So as you can observe here, the presence of horn. Okay. So if you see if the genotype is capital H, the male will have the if it is a female, the female will have the hen feather, the male will also be hen feather. But if it is a capital H and small h, both of them will have the similar character. But look at the condition when it is small h, small h. So when it is small h, the female will have the hen feather, but the male will have this cock feather. Okay, so although the genotype is same in both the sexes, but the phenotypic expression is different. That means the expression of this kind of character is only limited to one sex of the organism, that is only to the male. Then there is
Madam,
sex
is there any
influenced
cause for
character.
this particular reason?
Sorry. Oh, it is there. See, it could be because of the presence of other factors like hormones, etc. Okay, because the males have different set of hormones, the females have different set of hormones. So it could be influenced by the hormones as well. For example, you can take the means example of humans also. The development of breast, the development of beard, etc. So it is limited to one type of sex only. So that could be because of the influence of the hormone. So although the genotype will be same, but the phenotypic expression will differ. Then there is another thing called as sex influenced character. So influenced character means the similar to, it is somehow similar to limited but not same. So if you observe the baldness, have you ever seen a bald female? The baldness is quite common in males, right? But observing a bald female is quite rare. So if you see, when it is a homozygous dominant condition, both the male and the female can go bald. But if it is a case of heterozygous, the male will go bald, but the female will not be bald. So this genotype that is dominating in the male is becoming recessive in case of females. So this is influenced by the gender or the sex of the individual. So that is what is called as sex influenced character. Okay. Then comes a concept of penetrance and expressivity. So penetrance means in a population how far the genotype is penetrating. Okay. For example, if you see this multiple fingers, okay, the presence of multiple fingers, this is a genotype which is means if 100 people will be having this genotype, only 10 people might express the phenotype. That means the gene cannot penetrate completely in the population. So penetrance is actually a measure of the expression of the phenotype of a particular genotype in a population. So if you observe this first condition, okay, so the blue color is actually the expression of the phenotype. So if you see among 1, 2, 3, 4, 5, 6, 7, 8, 9, 5, 9. Okay, so among these 9 individuals, only 1, 2, 3, 4, 5. 5 individuals are expressing the phenotype, whereas all of them have the same genotype. So this is a case of variable penetrance, or we can say the gene is unable to penetrate completely. Now, what is meant by expressivity? Expressivity is the degree of penetration. Means even if every individual is expressing the genotype, how well they are expressing that particular phenotype. So if you look at the color pattern, so this should be the phenotype of the genotype. But if you look, everyone is expressing the phenotype, but the color intensity is different. Okay, that means the expression degree, the degree of expression of the genotype is different from individual to individual. So this is an example of variable expressivity. So penetrance means the measure of expression of the phenotype of a particular genotype, and the expressivity is the variable in the degree of expression. So in this condition, if you observe, there is variability in the penetrance as well as variability in the expressivity. Means not everyone having the genotype is expressing the character. So this is not expressing, this is also not expressing, this is also not expressing. though they all of them are expected to give a blue color, but they are not giving. So the gene is unable to penetrate into the entire population. But if you see into the expressivity, the degree of expression is different. Here it is very dark, here it is little bit lighter, here it is very light. So there is a variable degree of expression. So this is an example of variable penetrance and variable expressivity. Then comes the environmental modifications of gene expression. So sometimes whatever genotype you might have, that can be altered or that can be affected by the environmental conditions like the temperature or light, etc. If you take the example of these two rabbits, so both of them, they are having the same genotype, but depending upon it, which environmental condition they have raised, they have different type of pigmentation pattern. So when the rabbit is grown in a condition where the temperature is 25 degree or less, they will have this Himalayan pattern. But if they are grown in a area or a condition where the temperature is more than 30 degree, they will have this Himalayan coat color. Okay, so this is the environmental modification of the gene expression. So this is actually measured by a term called as phenocopy. So phenocopy means, simply you can remember like this, copying the phenotype. So both of means, for example, if you see there is a plant having capital T and small t. So what phenotype we are expecting from this? We are expecting a tall phenotype. But somehow if the plant is not getting enough nutrition or if it is not getting enough sunlight, so it may not grow as tall as we are expecting. So it will be appearing dwarf. But actually it is having the genotype of a tall plant, but it will appear dwarf genotype. So that means due to nutritional effect or due to environmental effect, it is copying the genotype of a dwarf plant. So that is what is called a phenocopy. Okay. So the phenocopy can be observed in Drosophila also. The Drosophila, if they are grown in high temperature, so they will have a phenotype of the wrinkled, means whatever wing they are having, the wing will be somehow defective. So the defective wing can also develop because of a gene mutation in the Drosophila. But in a wild type Drosophila that is grown in high temperature environmental condition, it will also have a similar phenotype like that of a mutated phenotype. So this is what is called a phenocopy. So the copying of the phenotype of another genotype. Then coming to the fine structure analysis of gene. Okay, so actually gene, the modern concept of gene can be expressed by three different ways. Gene as a unit of cistron, okay. Gene as a unit of recombination or recon, gene as a unit of mutation or mutant. So we can express gene as a unit of transmission. So transmission means the cistron refers to the portion of the DNA that specifies a single polypeptide chain. Or gene can be explained as a segment of DNA that codes for a protein. So we can call it a cistron. Similarly, gene can also be expressed as a unit of recombination, or you can call it as recon. So recon is the name of the tiniest piece of DNA that may be that may be detached and exchanged with another chromosome. That means which can undergo crossing over, okay, or which can undergo a recombination process will be called as a recon. Similarly, the segment of DNA which can undergo a mutation, so that can be also called as a gene, which is mutant. So these are the modern concept of gene. Now, there was one scientist called Simon Benger who actually gave rise the concept of gene through his experiments with a bacteriophage and the ability of the bacteriophage to cause the plaque. Now, what is mean by plaque? So, if you see, everyone know what is a bacteriophage, right? So, bacteriophage is actually a virus that infect and kills the bacteria. So, there is a bacteriophage called as this bacteriophage. If you see, so it has a certain gene called as a R2. So, R actually this R2 is the segment, or this is the gene which is actually responsible for lysing the bacterial colonies. So if you co-culture a bacteria and if you infect it with a bacteriophage and you grow it in a agar medium, what the bacteriophage will do, the bacteriophage will go inside the bacteria and will kill it. So because of the killing of the bacteria, what you observe in the plate, there will be holes like this. Okay, as you can see. So these holes are called as your plaques. So the wild type bacteriophage that has the normal gene actually forms very small plaque like this. Okay, and the plaques are not clear because it cannot lyse the bacteria completely. So it will have small plaques like this. But if there is a mutation in the gene that is responsible for lysing the bacteria, then you will have large plaques like this. And the plaques will be very clear or transparent. You can actually see in the agar plate that there are small transparent areas have been created. So these are called as the R2 mutants. So the R2 mutation in the bacteria, the R2 mutation in the bacteriophage is responsible for lysing the bacteria very rapidly and creating very transparent and large plaques like this. Okay. So what he did actually, now he took two different type of mutants. So as you can see, there is R2 mutant 1, there is R2 mutant 2. That means the type of mutation in mutant 1 and mutant 2 were different. Now, what he did, he co-cultured or co-infected both the mutant forms into a bacteria that is E. coli B. So in E. coli B, what happened? If the mutations are like this, okay, you can look into the bottom part of the diagram. Okay, so if the mutations were located on the same gene like this, the gene one is mutated in both the cases. So that means the gene one is completely non-functional. So with a single gene 2, here if you observe carefully, in the first case the G1 there is a mutation in the gene 1, and in the second case or in the second gene there is a mutation in the gene 2. So if you combine both of these, so here the gene 1 is non-functional, for this copy gene 2 is non-functional, but if you combine them you get a complete functional gene. Gene 1 is also functional, gene 2 is also functional. So in this case they can behave as a wild type bacteriophage. Okay, but if the mutation is like this in the second case, that means in the mutant 1 also the gene 1 is mutated, and in the mutant 2 also the gene 1 is also mutated. So in this case the gene 1 is completely mutated. So only the gene 2 cannot lyse or cannot form the plaques in the bacterial cell. So what conclusion can be drawn from this? Now, if the genes are or the mutations are located on different genes, they can recombine to form a restore a wild type character. So here in this plate, after the co-infection in the E. coli B, all possible type of plaques can be formed. But if you observe in the E. coli K lambda, so this is a different strain of the bacteria. So this can only be lysed or this can only be infected by a wild type. So when the wild type will be possible? Now when the mutation is like this. Okay. So this was again confirmed by a C-Strass complementation test. Okay. So what he did, as you can observe, I think the slides are a little bit smaller. Is it visible to you all? Means you can all understand what is written in the diagrams.
Yes,
The
ma'am.
diagrams, are they visible? I don't know how to zoom in. If it can be zoomed. Okay, let me try to zoom it actually.
Mobile exam ହେଇଯାଉଛି madam.
Okay, you can zoom it, right?
Yes, ma'am.
So just focus on this part. Okay. So what he observed, now if there is mutation in two different genes like this. Okay. So here the gene 1 is mutated, and in the other case the gene 2 is mutated. So they can actually complement each other. So because you are at least getting a functional copy of A and functional copy of B. But if the mutation is in the same unit, so from here actually the concept of, he gave the concept of cistron and the concept of recon, that is the unit of recombination. So here the genes, if I cross it like this, so what you will observe, there will be a double mutant and there will be a completely functional gene like this. Okay. So in those in those bacteriophages where the mutations are located on two different genes, they can be complemented each other or they can complement each other. But if in a particular bacteriophage the mutations is present within the same gene, so they cannot complement each other. So there is no complementation. So this is what is called as the fine structure analysis of gene. That means as long as the mutations can be complemented, the mutations are part of two different units of recombination. But if the mutations cannot be complemented, they are part of the same cistron or they are part of the same recon. That means they belong to the same gene. So here if you observe, there is no lysis in the E. coli B because the mutation is present in the two different locations of the same gene. So this was the concept given by Simon Benger in the bacteriophage. Okay. So I hope this was clear to you. Simple thing. If the mutations are located on two different genes, the other functional copies can actually complement each other and you can observe the plaques are there. That means the bacteriophage is functional. But if the mutations is occurring within the same gene, then no complementation will occur and you cannot observe any kind of plaque in the bacterial phage. So this is what is called as the cis-trans complementation test. So if the mutation is trans, means they are located on two different segments of gene, then the complementation will occur. But if the mutation is cis, that means they are present on the same gene. So there will be no complementation. Then coming to the more important parts actually, that is linkage and crossing over. So if you observe this particular cross, that is similar to a Mendelian dihybrid cross. Okay, so here also two characters are taken. One is the purple color flower with the long pollen, and another is red flower with the round pollen. So when this kind of dihybrid cross is done, so we should expect a phenotypic ratio of what? 9:3:3:1. But what was observed, the phenotypic ratio was something different. So what was the ratio? Now the ratio was somehow 14:1:1:5. So this is complete violation to the Mendelian dihybrid cross. So why this kind of thing is happening? Now this is because of a phenomenon called as the linkage. Now what is meant by linkage? Now when on a chromosome two genes are present very close to each other, then they will be inherited as a single unit during the meiosis. That means if the two genes are close enough, they will not be separated by crossing over. So this concept is called as your linkage. So in case of linked genes, the percentage of recombination will be very less. So there is one thing which you must remember, that is the maximum percentage of recombination. So the maximum percentage of recombination will be always 50%. That you can observe in case of independent assortment. So in Mendelism, if you remember, the percentage of in case of dihybrid cross, the maximum percentage of recombination that can occur is actually 50%. That means 50% will have the parental combination and the 50% will have the recombinant character. So here, if you can observe, look into the first example. Look at the location of the gene. Okay, so the gene, that is whatever character it is coding, yellow and white. So the genes are actually very close to each other. So when the chromosomes will segregate, the both the character will together go in the same chromosome. Okay, so in this case, the parental combination will be 99%, and the recombinant is very less, that is only 0.5%. But if the genes are actually far away from each other, or there is a distance between the genes, then they can undergo crossing over like this. So they will have 50, let's say here the character is actually 65% is parental and 35% approximate is recombinant. So depending upon the distance between the gene, either the genes will exhibit linkage or they will exhibit recombination or crossover. So the lesser is the distance between the gene, the higher will be the linkage. See, it is very simple. If two genes are very closely located to each other, so during the chromosome breakage, they will not be broken and they will never be separated from each other. So both the character will appear together always. But if the genes are located far away from each other, so during the meiotic crossover, the chromosome will break from this point and the chromatids will be exchanged, and this will lead to development of new character combination. So we will see some examples, then you can easily understand. So this is how exactly the crossing over occurs during the meiosis. Okay, so these are the centromere. These are the points of chiasmata where actually the chromatids will break and the parts will be exchanged. Okay, like in this case. I hope this is visible to everyone. So here, if you see, two characters are being considered. One is long wing with gray body. Another is vestigial wing. Vestigial means small wings. Okay, small wings with black body. So look at the genotypic combination. So here VG plus stands for your long body, long wing, and B plus stands for a gray body. So VG, it stands for vestigial wing, and B, small b, stands for black body. So in the parent generation, you are having a homozygous genotype. So in the F1, this parent will give a gamete that is VG plus and B plus. This parent will give a gamete that is VG and B. So when they will combine in the F1, what you will have a heterozygous genotype VG plus B plus from this parent, okay, this chromosome, and VG and B from this parent from this chromosome. So this is a F1 hybrid that will have the dominant parent phenotype, that is long body, long wing and a gray body. Now, when you will cross it with a homozygous recessive parent, okay, so this can have 4 different type of gamete. So if no recombination will occur, simply this chromosome will be there and this chromosome will move here. So there is no recombination. But if recombination occurs, that means here exchange of parts will take place. So carefully observe this first diagram. Here there has been crossing over. So that is why in the VG plus you have the B, and here with the VG plus you have got the B. So here you have VG with the B plus, and the complementary you have here, and this gamete is donated by this recessive parent. So accordingly you have 4 different characters. So long wing and gray body like the parent, vestigial wing and the black body like this parent, but the vestigial wing with a gray body, that is a recombinant character, and long wing with a black body. That is again another recombinant character. So how you will predict the map distance between them? Now simply the formula used is the number of recombinants divided by total number of progeny multiplied with 100. So if you observe here, the total number of recombinant is 92 plus 88, that is 180, divided by total number of progeny is actually here 1000. So 180 divided by 1000, that is your so 180 divided by 1000, that is your 18. So what is the distance between these two genes? The distance between these two genes can be expressed as 18 map unit or 18 centimorgan. Okay. So this is an example of a two-factor cross, because we are considering only two gene loci, or two gene loci are considered at a single cross. Then similarly we have three-factor cross also, like this. So in the three-factor cross, first let's understand what is meant by cis position, what is meant by transposition. So when all the dominant character of a allele are present on one chromosome, that is a cis position. And when all the recessive characters are present on the other chromosome, that is a cis condition. Okay. So here all the dominant genes are present on one chromosome, all the recessive genes are present on one chromosome. So this condition is called as cis position. Cis means same. All dominant alleles are on one chromosome, all recessive alleles are on the other chromosome. So here plus means a dominant character or wild type character. But if you observe here, two dominant characters are there, but this is a recessive character in the middle. So the position of A and C are actually cis because they are on the same chromosome. But the position of A and B is trans, and the position of B and C is also trans. And here the position of B and C are cis, but the position of A and B are trans. The position of A and C are also trans position. So this is what is called as a cis position or coupling, and this is what is called as a transposition or repulsion phase. So this is actually very important when you are considering the three-factor cross. Now, what kind of problems you can get in the exam? So, if you look here, the crossover is being done here. So, there is a 3-point crossover actually it is. So, three different genes are considered. One gene is SC, the second gene is EC, the third gene is CB. So, these are the recessive forms of the gene, and this SC plus, EC plus and CB plus are the dominant form of the gene. So if you cross these two types of, so the what is the characters? The character is skewed bristles, that stands for SC. Then echinus eyes, EC. Then cross veinless wings, that is CB. Okay. So this is a wild type, means it will have normal bristle, it will have red eye, and it will have cross vein wings. So that is simply summarized as wild type. So if you observe a cross like this, so this parent will give this gamete combination, and from this parent you will have this gamete as well as this chromosome. So this chromosome, this is a Y chromosome. So you will not get any multiple combinations here. But here, as each of the gene pair is heterozygous, you can have different recombinations here. So if you look into the problem, whatever is mentioned here, you can have different numbers like this. So this phenotype is what? Skewed, echinus and cross veinless. And what is the number? 1158. Then wild type, so what is the number? That is 1455. So these are actually the number of gametes. Okay, these are the number of gametes that will be produced. Now, what are the possibilities here? Now, one possibility is there could be a crossing over between these two genes, or there could be a crossing over between these two genes or there could be a double crossover, means simultaneously two crossovers between these two genes at a time. So these are the genotype of single crossover. Okay, let me draw. Actually, with the drawing, this is very difficult. So, is the drawing visible to you now if I'm drawing like this?
Yes, ma'am.
This red ink is visible, right? Okay. So let me show you. See, what are the possibilities of this condition? So there could be a single crossover between these two. So if there is a single crossover between these two genes, that is SC and EC, so what you can do, now this will go to this chromosome, this segment, and this SC will come to this. So what will be the new genotypic combination? Now there will be SC plus EC and EV. So where you can see this one, SC plus EC and EV. If there is a crossing over between SC and SC and EC. Similarly, there could be another crossover that is between EC and EV. So in this case, what will happen? The CB plus will go there and the CV will come to this side. So what genotype you will have? You will have SC, EC and CB plus. That you can see here, SC, EC and CB plus. Okay. So this is another genotype. So these two are the parental genotype. This is the crossing over between the gene SC and EC. Okay. And this is the crossing over between the gene EC and EV. And these are actually the product of double crossover. So let me draw a simple thing and then you will be able to follow. So I am using eraser. So let me draw a simple thing, then you can follow me. So see, this is a condition. Okay, I'm drawing. This is A plus, this is B plus, this is C plus, and this is A, B, C. So what possible things can occur? Now there could be a single crossover between A and B. So if there is a single crossover between A and B, so what genotype you will have after the crossing over is done? Now this A will move to this part and this A plus will come to this one. So the new genotype will be what? A, B plus and C plus. And what is the other genotype? That will be A plus, B and C. Similarly, if a crossing over will occur between B and C, so what genotype you will have? The genotype will be A plus, B plus and C. Then it will be A, B and C plus. Right? Everyone understood this part or not? So these are the single crossovers. Now, if there will be a double crossover, so double crossover means what? Now, at a time, there will be crossing over between A and B, as well as there will be crossing over between B and C. So in this condition, what genotypes you will get? You will have A plus, B and C plus. Then you will have A, B plus and C. So these are the product of double crossover. So now how you will identify it in a problem that is given? So I will actually use this problem to make you understand. Now here you have a question, right? So if you see carefully, what is the highest number that you are observing in this table? Now the number is 333 and 340. So it means these two are the parental genotype. Because if you observe here, if no crossover will occur, the chromosomes will move like this. Okay, so there will be means most of the gametes will have only this genotype. Either they will receive plus plus plus, or they will receive SC, EC, and EV like this. You are observing, right? So the maximum number of whatever maximum number of progeny that you are observing, that is actually a parental combination. And what is the lowest number that you are observing here? The lowest number is this 4 and 4. So this means these are the product of double crossover, because the means two crossover to occur simultaneously is not a simple thing. So usually the two crossovers will be least in number. So from this you can actually identify what is the parental genotype and what is the double crossover genotype. And by comparing the parental and the double crossover, you can actually predict what is the order of the gene. So like I will be using this one. So yellow, vermilion and cyan means what should be the genotype here? For yellow, what is the symbol used? Small y. For vermilion, the symbol used is V, and for cyan, the symbol used is SN. Okay. So, but if you look into the parental genotype, that is the 333, what is the sign? Now that is having Y, SN, and V plus. Okay, because vermilion stands for V. Since no V is present, it means it will have a V plus character. Then what should be the this one? So this one should be Y plus SN and V. So in all of you understood this part or not? So how to assign the genotype? Okay, you can ask me if you're not following. I will try to explain as much as possible, but at least you should let me know. So this genotype is actually, since it is yellow, vermilion and cyan, as per the question that is given, the genotype we should assign is Y, V and SN. And what should be the genotype of this one? Now yellow, so Y is there, vermilion, V is there, but since nothing is mentioned regarding SN, so it must be SN plus, or I am writing it simply S. Okay, so here this is Y, V and S, and for this it is Y, V and S plus. Now for this it is Y and nothing is written, so it is V plus and S. And this genotype is Y, V plus, S plus. Then this genotype is vermilion and cyan, so that means it is Y plus, then V and S plus. Okay. Then here, since it is only vermilion, means it is Y plus, V and S plus. Only cyan means it is Y plus, V plus, S. This is wild type means what? This is Y plus, V plus, S plus. Okay. So if you look into the number, what I told you, in any kind of problem, if you see the maximum numbers and that are close to each other, because 333 and 340, they are almost same in number. So this will represent the parental. So what is the parental genotype here? Now that is Y, then V plus and SN. And what is the other one? Now that is simply complementary to this. If this is Y, the other parental genotype will be Y plus and V and SN or S plus. Just let me take it as S. Okay. Now I told you what is the double crossover. So the double crossover is actually what is the lowest in number. So what is the lowest number? These 4 and 4. So what is the genotype of the double crossover? Now that is Y, and nothing is mentioned, means that is the plus. So B plus and S plus. Okay. Then the other one is B and S is there. So this is Y plus V and S. Okay. So this is the genotype of the double crossover. Now, if you compare a wild type or a parental combination with a double crossover, so what is the parental combination that we have here? That is Y plus V and S plus. And what is a double crossover? Double crossover is Y plus V and S. So now, if I tell you, so here the Y and S both are positive, that means they are in a cis position. Okay. In the parent. Okay. Now the V and S, they are actually in the trans position. The Y and V, they are again in a trans position. But if you look into the double crossover, the Y and V, they are in trans. So earlier also, the Y and V were in trans position and the Y and S were in cis position. But now if you see, the V and S, they were in trans position, but now V and S are in cis position. Similarly, the Y and S were in trans position, but now the Y and S are in, Y and S were in cis position, but now they are in trans position. So if you observe, the S has changed its position with respect to V as well as with respect to Y. So this means the order of the gene is actually like this. The gene Y, then S is in the middle, and then you have the V. Okay, so what is the parental combination here? I think that diagram is not given, but you can means do it on your own. I will explain this part. Then this example you can do it as your homework. Okay. So here, if you see the whatever problem is given in the example, that we will consider. So here the problem is little simple because the order of the gene is actually correct. So as you can observe here, whatever we discussed, so this is the actual order of the gene. So if there is a single crossover between the gene SC and EC, see, this SC part will move to this chromosome and this part SC plus will come to this chromosome. Okay, so here what is the number that is given in the question? That is 163. This is 130. That is already given in the question. Similarly, if there is a crossing over between EC and CB, then what is the new genotype that you will have? SC, EC and CB plus SC plus EC plus and CV. And what is the number? 192, 148. Now, what is the genotype of the double crossover? So, double crossover means this part will go to this location, this part will also go to this location. So, both the parts will be exchanged. So, now if you see SC, actually this chromosome is this one. So, EC is there, but it has got SC plus from this and CB plus from this location. Similarly, here the EC plus is there in the middle, but CV has come to here, okay, and SC has come to here. So what is the number of these chromosomes? Now that is simply 1 and 1. So whenever there is a crossover between the single genes or single crossover is happening, at the same time a double crossover is happening between these two genes as well. So here also, when there is a crossing over is happening between these two genes, again a double crossover is happening between these two genes. So what is the total number of crossover, or how to calculate the distance between the two genes? Now simply by adding the total number of recombinants. So the total number of recombinants is 163 plus 130 plus 1 and 1, that is 2. The total number is 295. And here, what is the total number of recombinant between the EC and CB? The total number of recombinant is 342. So how to calculate the map distance between the gene SC and EC? The map distance is 295 divided by 3248. The 3248 is nothing but the total number of progeny that you have obtained. Okay. Similarly, here, what is the total, the map distance between the EC and CB plus? Now, the map distance between the EC and the CB plus is 342 divided by total number of progeny multiplied by 100, it should be, that is 10.5 centimorgan, or you can call it as map unit as well. Okay, so this is how you will be calculating the distance between the gene using the recombination frequency or using the percentage of crossing over between the two genes. Then there are two different new concepts. One is called as the coincidence. The other one is called as the interference. So coincidence is the ratio between the observed number of double recombinant divided by the expected number of double recombinant. So if we go back to the same problem, here the observed number of double recombinant is how much? 2. Only you are observing 2 number of recombinants, 1 and 1. But what is the expected number of double recombinant actually in this case? The expected number of double recombinant is actually you have to multiply both the recombination frequencies. So both the recombination frequency is 0.0091 into 0.105. This value multiplied by 3258. Actually, the number will come somehow. So the number will come somehow 31. Okay. So you should be expecting 31 number of double crossovers, but actually you are observing only 2 number of double crossover. So what is coefficient of coincidence? That is expressed by C is actually 2 by 31. Or in other terms, they have expressed is that the recombination frequency, that means 2 divided by 3248. The value will be same as this one. Okay, 0.0006. So 2 by 31 will be also giving you the same value, that is 0.063. And what is interference? Interference is 1 minus coefficient of coincidence. So here the coefficient of coincidence is 0.063 and the interference is 1 minus 0.063, that is 0.937, or we can say 93.7%. So what is actually interference? Now interference is that when 2 crossovers are happening, the first crossover is having an inhibitory effect on the second crossover. Okay, so if we go back to this slide, as you can see here, 2 crossovers are happening simultaneously. So the first crossover is actually having an inhibitory effect on the second one. So if the interference is more, as you can see in this case, so there is 93 or 94% interference is there. That means 94% times the first crossover is not allowing the second crossover to happen. So if the second crossover will not happen, then you will not have any double crossover. So that is why where you are expecting actually there should be 31 double crossover progeny, the actual number is very, very low, that is only 2. Okay. Now this is another example of means the same how the map distance can be calculated by using the pedigree analysis. So in this case, we are observing or we are considering linkage, and the linkage is used in the pedigree chart to expect the number of the distance between 2 genes. So here we are observing 2 different characters. One is the blood group. The blood group is B, and the other character is the nail patella syndrome. That means the individual who will have a blood group B will have defective nails and defective knee. So whenever the blue color is actually representing the individual having the nail patella syndrome. So now if you see in the second generation, sorry, in the third generation, most of the blood group B have the nail patella syndrome. But if you look carefully here, okay, this is having a blood group B, but it is not having the nail patella syndrome. Similarly, this is having a blood group O, but still having the Nel patella syndrome. And here also, this is a blood group B, but not having the Nel patella syndrome. So, this is something different from the parental combination. So, these are actually nothing but your recombinants. Okay. So, here also, this is having a blood group B, but it is not having the Nel patella syndrome. So, among a total number of 13 progeny, only 4 are having the recombinant character. So what is the recombination frequency? So that is 4 by 13, or it is nearly equal to 31%. So what is the distance between the gene of blood group B and the gene causing the Nel patella syndrome? Now that is simply 31 map unit, or the recombination frequency between them is actually 31%. So actually what we are doing here, how we are calculating the map distance, now, as I told you in the previous slide, the more is the distance between the gene, the higher will be the frequency of crossing over. And the less is the distance between the gene, the less will be the frequency of crossover. So in any kind of cross, if you are observing that the number of recombinants is more, then the distance between the genes should be more. Or we can, in other terms, we can say the distance between the genes is actually represented by the crossover frequency. So we are using this crossover frequency to predict the distance between the genes. So more is the crossover frequency, the more is the distance, the less is the crossover frequency, the less is the distance. So if we compare between these two, actually these two conditions, here the distance between these two genes is 9.1 centimorgan, because if you see the number of recombinants is less than this one. 295 is less than 342. So if you are actually placing the gene like this, so the gene should be SC, the EC should be little bit closer, and the CB should be little bit farther than the EC. So this distance is 9.1 centimorgan, and this distance is 10.5 centimorgan. Okay, and what is the distance between these two? You can simply add them. Okay, so that is how you can find out the distance between the gene by using the crossover frequency. So actually this is a little bit difficult to explain without drawing much, but I have tried my best. I hope you have got few things, and if you have any queries or any doubts, you can ask me. If you go through the material 1 or 2 times, then you can easily understand it. So that is all from my side. If you have any questions, you can put, or we can end the session if there is no question.
Madam, is there any particular timetable for online classes?
See, I think they might have given you a schedule. Every time it is scheduled from 6 to 8 only. And what as per my knowledge, actually continuously 4 days are given to one counselor to complete one paper. So like today I completed your block 1. So tomorrow, day after tomorrow, and next day after tomorrow, I'll be completing the next 3 blocks. I think they
Oh, that
have
means
shared something.
consecutive 3 days.
Yes, for consecutive 4 days, you will
Oh,
be attending one paper.
from 6 to 8 p.m.
Okay, can you
Is there
repeat the
any
question?
timetable of offline classes?
Offline, no. Actually, these things you can ask to the management team. Actually, we don't deal with this. We only will be dealing with the academic part. So, if you have any questions or any queries related
Okay.
to means teaching or anything, you can ask. The rest of the things you can contact the regional center. Okay. Okay, for 5 marks, see,
Okay, madam.
actually, there is no limitation as such. Whatever question is being asked, you try to cover up that question only. It doesn't matter how long or how short you are writing. If your content is means your answer is having the content of the question, that should be enough. Okay, any other questions?
Could you please tell some more words about Nel patella syndrome?
Actually, the nail patella syndrome, that is a genetic defect. If the syndrome is actually the development of the nails. Okay, that will not be proper. Means you will have defective nails, and you will have some problem in the patella. Means the knee bone actually. It is. Okay. So there will be certain problem in that only. It is a genetic disorder actually. So there will be some kind of abnormality, physical abnormality will be there. So
Okay,
usually it is
okay.
observed that any individual having a blood group B will be having the nail patella syndrome. But it is not like fixed, but the genes are actually correlated. So whenever an individual will inherit the blood group B from the parent, it will also inherit the defective gene of the nail patella syndrome.
Oh. That means they are linked genes.
Uh, Actually, yes, the genes are actually linked. So whenever the blood group B will be there, the gene for the defective nail patella gene will also be there. Okay. Yes, I can provide you with the
Okay, thank
PPT.
you.
The all the PPTs means everything. The all the content are from your material only. So if you have that e-Campus material, everything I have adapted from that only. So I suggest all of you to go through this PPT or material, whatever it is. And if you are having any queries regarding this, I can also try to means solve it tomorrow. Also, I will give some time for this. So you can take this. So from this, okay, what kind of problem you have on one gene, one enzyme hypothesis? It simply means it simply states that every enzyme is coded by a particular gene. Okay, so if you are having one gene, one enzyme, that enzyme must be coded by a particular gene. So how that was actually detected? Now, if there are multiple genes in a pathway, and if you are somehow mutating a single gene, then the entire pathway will be interrupted because that gene is defective, that enzyme cannot be synthesized, so it will lead to a defective phenotype or defective protein. So it will interrupt or it will inhibit the pathway. So one gene, one enzyme simply predicts, or it simply proposes that every enzyme is coded by one particular gene. Okay, anything else? Okay, regarding the PPT, I don't know where I can share it, means where to send it. I have no idea where the common things can be sent or anything. So, if one of you can write your email ID, I can send it, and the rest of you can, you can circulate it among yourself. So, if anyone is interested, you can just send me your email. You can write down your ID, I will try to send the PPT to you. Whoever is asking. Okay. Manoranjan 9853. Okay, just wait. Okay, I will do one thing. I will share it with the group actually, means whatever platform we are having. Then you can means take it from them, or I will ask the headquarter to share it with all the learners. Okay, that will be better. Regarding the discussion, actually these things you should means discuss with the management. If they can create any platform, we can discuss, there is no problem. You can request them to create a platform. Okay. Yes, I do agree and I do understand, but that is not exactly in our hands. So you can request the management if they can create a WhatsApp platform or anything where the learners and the counselors can interact. That will be better. So they can do it. Okay. Okay, fine. Please try to go through the whatever we discussed today, and let me know if you come up with any doubt. I will try to means discuss something tomorrow also. Okay, I will try to finish tomorrow class a little bit early, and if you have any questions from today's class, we can discuss tomorrow. So thank you all for your patience. Okay, happy learning. So I'm signing off. See you tomorrow. Okay. Okay, fine. Thank you all.